Bom, pessoal, boa tarde a todos. Agradeço a presença de quem está nos ouvindo hoje. Hoje vamos ter, então, o prazer de falar um pouco com o Michel Kulbicki. Ele agora está aposentado na França, mas ele continua ativo em diferentes âmbitos da biogeografia marinha. E hoje a gente vai começar a fazer, aos poucos, um esquema diferente, além do palestrante, convidar outro pesquisador para ajudar a mediar a discussão que a gente está tendo ao longo de do nosso CEBIMário. Isso vai permitir que nossas discussões sejam ainda mais nutritivas e tenha muito mais caldo para a gente discutir. Então, nesse sentido, eu convidei hoje a Mari Bender. Obrigado, Mari Bender. Mari Bender é professora da Universidade Federal de Santa Maria. E temos, então, a presença do professor Michel Kulbicki, que, por acaso, a câmera dele não está funcionando, então ficará a tela preta. Peço desculpa a todos, mas o Michel tentou, mas hoje não deu. Então, thanks, Michel, for staying today here. It's very exciting, your presentation. And, well, I think you try to sharing your screen and start your presentation.
All right.
Maria, você precisa depois parar a sua câmera e pôr lá em mute.
All right, so everything is fine here?
Sim, Michel, tudo ok.
Okay, so let's start this presentation. Well, hello everyone. What I'm going to talk to you about today is diversity, and it's linked to biomass, production, density, and so on. And we're going to take the example of reef fish, and mainly reef fish from the Pacific Ocean. So what's the major question we're going to look at today? It is a long-standing debate that to know if the biomass production is or is not linked to diversity in some sense, and in particular to species richness and functional richness. Okay, if such a link exists, it is likely to be governed by the same factors which rule species or functional richness, so we have to check that. But there are a number of methodological problems linked to this question, and in particular, it's quite difficult to measure richness, biomass, production, abundance in open systems, in particular reefs. Measuring the factors associated to these parameters is also a bit difficult. And last, we need to sample areas with contrasted values for the above factors and parameters, and this means quite large data sets. So today what I'm going to present you is preliminary, but please feel free to use anything presented today. So we are going first to have data which we could call regional diversity. That is, we know that there are more than 6,000 species of reef fish across the world. And we have at present available more than 250 checklists of these species. Just to give you an idea, this is an old map, it's 2012, and it had only 165 checklists at the time. So you see that we do have quite a nice coverage of reefs across the world. So for most species, we do have a number of life history traits, such as diet, size, and so on, which is available for most species. Then we do have other traits such as pelagic larval duration, growth, depth range, and so on, which are available for only a fewer number of species. Then we have all kinds of factors which can be linked to these species, in particular physical factors such as island size, reef size, island isolation, and so on. And also biochemical factors such as salinity, sea surface temperature, and so on, and human-related data such as human density, MPAs, and you name it. So what we, with all these checklists, what we've done is to calculate the number of species which in 5 by 5 degree cells on a worldwide basis. And for each of these cells, we'll have the species composition, the distribution of the life history traits, the physical and biochemical factors we talked about, the human associated data, and also what we will call functional composition. We'll talk more about it later. So we also do have field data. In particular, this is the observation of reef fish directly on reefs. And we have data from a number of places across the world, and the data set is still growing. But today, I'm going mainly to look at a very small area. Well, small. It's more than 6,000 kilometers wide, but a restricted area in the Southern Pacific to illustrate the concepts I want to talk to you about. So for each of these transects, we have 12,000 transects altogether now. We do have the species composition, the abundance for each species, the size distribution of each species, and also data on the landscape. So with abundance and size distribution, we can make up estimates of biomass and what we will call production, but we'll talk about this a bit more later. Now, results. Let's first look at how the various levels of diversity link to one another. First, gamma diversity, or what we could call regional diversity, and alpha diversity, which we could call local diversity, usually are related by an asymptotic curve of the kind you see here. So to check this, we looked at our study zone, in which we do have 9 regions, which will determine what we'll call gamma diversity at the regional scale. Then we have 32 islands for which we'll have diversity at the island scale, and then a certain number of transects for which we do have a local diversity, in other words, the number of species seen on a given transect. Now, if we look at the correlation between regional diversity and diversity at the island level, we do see that there is an increasing trend, but that we do also have very strong variance within a region. Now, if we look at the next scale, that is between the region and the local scale, that is on a reef, you do see also that there is a positive correlation, and that there is a low variability within a region and a strong variability between regions. Now, the last correlation we can look at is the diversity at the island level with the diversity on the reefs. And you do see again that there is an asymptotic curve, and that you get to some type of saturation when the diversity on the island gets very high, and this is linked to an increase of beta diversity. Then we can look at the relationship between species size and richness. Okay, the species size within a region and within a reef is correlated to the richness at the local level, island level, and regional level. Let's look at this. So from the metabolic theory in ecology, or MTE, developed by Brown, we know that as species richness increases, the average size of species tends to decrease. So if we do look at this on a global scale, here we do have the proportion of small species, that is less than 8 centimeters as adults. In gray, you have a null model, in other words, a random distribution of these species according to species richness. And in orange, you have the observed values. And what you do see is that there is an increase of this proportion as the species richness at the regional level increases. Each point representing a different region, by the way. Now, if we look at larger size classes, you do see that little by little, we're getting to the same thing than the neutral model. And if you go beyond 30 centimeters, it's the opposite. You do see that there is less and less large species as in proportion as the species richness increases. So all this is okay with the expectations of the metabolic theory. Now, if we look at the local level, we're having strange results here. So this the species richness at the local level, that's in other words, the number of species on a given transect. Each point here is a given transect. You have about 1,500 points. And here is the proportion of large species. And you do see that there's kind of an increase. Now, if we do look at the average individual weight, as species richness increases at the transect level, you do see also that it increases. And both of these results are opposite to expectation. Unfortunately, at present, we do not have an explanation for this phenomenon. Now, if we look at different regions for the same parameters, that is, the proportion of large species or the proportion of, the average individual weight, in red you have French Polynesia, which is a region with low gamma diversity. And in blue, you have New Caledonia, which is a region with high gamma diversity. And you do see that indeed in a region with high gamma diversity, you do have on average less large species and smaller species, individuals, sorry. So this is again okay with the metabolic theory. Now, if we look at species richness versus large-scale factors, we built a model with about 9 or 10 factors. We're going to see this a little further. And we do see that the correlation is extremely good between observed richness and predicted richness. Now, if you look and at the maps of observed versus predicted, you can see that the two maps are nearly identical. So what were the major factors we looked at? We looked at factors which we can split into 5 different hypotheses: the island biogeography or area hypothesis, evolutionary history, energy hypothesis, in other words, the role of temperature, connectivity and hypothesis, in other words, how far you are from a source, and the mid-domain effect. And what you do see here is that the two major hypotheses are the island biogeography and the evolutionary history, which can predict most of what we do see in terms of richness across the world. Now, we talked about so far species richness. What about functional richness? Okay, so we know that species richness is related to functional richness from a number of theoretical articles, and there are several ways to look at this. We can think that each species has one function, and in which case there is a one-to-one limit. In other words, you cannot see places where you have more functions than you do have species. Now, actually, a given species may have several functions. In particular, a small fish may have different function than the adult within a species. Sorry, it went a bit fast here. No, it's okay. Now, let's suppose that we have one function per species. And if one way we do simulate functions is to look at the combination of life history traits. This is only a proxy of functions, ecological functions, and therefore you should be very careful not to equate both. But it's a way to have some idea of what's going on in terms of ecological functions. Here on this graph, you see a combination on the lowest curve. It's combining 2 life history traits, then 3, then 4. And you can see that little by little you're going to this one-to-one limit. And if you had an infinite number of life history traits, this is where you would get to. But actually, for a given function, you may have several species. And if we do plot the species richness versus functional richness in terms of the number of species within these functions, We can separate functions where we do have only one species representing this function. In other words, we usually call these functions vulnerable functions, because if the species disappear, the function disappears. And then you have redundant functions in which you have a certain species redundancy. In other words, you have several species which have the same function. And you do see that as species increases, you do have a faster growth of the redundant functions versus the vulnerable functions. Now let's look at the map of total richness, which you have already seen, and functional richness. You can see that both of them are just nearly identical. In other words, the factors we looked at earlier for species richness are the same that control functional richness across the world for reef fish. So now let's go to the relationship between these richness, either functional or species, with density or abundance, whichever you want to call it. That's the number of fish per square meter of reef. We do expect some kind of exponential curve. If we do look at true data from the field, here we have 4 regions, 19 islands within these regions we analyzed. And you have the gamma diversity for each of these regions written in there. Now, here is a first example of Polynesia, which has a low gamma diversity, approximately 600 species, versus New Caledonia, which has about approximately 1,200 reef fish species. And you do see that in both cases, you do have an exponential curve, but that it grows faster in Polynesia than in New Caledonia. But you also do see that the average value for French Polynesia is lower than for New Caledonia. In other words, the density increases with richness, which is okay and expected, and there is a higher density for a given richness in species-poor regions compared to species-rich regions, which is rather unexpected. It is quite often asked in this type of analysis to replace species by fixed number of individuals per transect, and the results are exactly the same. So there are differences according to gamma diversity. First, in the number of species per transect. Here you have New Caledonia, Fiji, Tonga, and Polynesia, which are ranked by decreasing gamma diversity, and you do see that the number of species per transect tends to decrease with this diversity. And it's the same for density, which also tends to decrease as you go from species-rich to species-poor regions. So now let's go, the next step is to look at biomass versus richness relationship. So if we combine the previous results, that is, we have the average weight of species grows faster as local richness increases in low gamma diversity regions versus high gamma diversity regions. And we know that the density increases in the same way, but on an exponential curve. So from this, we would expect this type of relationship. In other words, the biomass should increase exponentially with the local species or the local functional richness. Okay, such curves have already been demonstrated in several articles, but I'm going to show you regional comparisons. So here you have the biomass. Be careful, this is a log scale. And French Polynesia, a region with a low diversity, and New Caledonia, a region with high diversity. And you can see that for a given species richness, you do have a higher biomass in Polynesia than you do have in New Caledonia. Now, well, this is, you have similar results if you with the number of individuals per transect. And if you look at the regional effect for biomass, you see that biomass tends to decrease with regional, on average, between a regional, as gamma diversity decreases. And we also here, you can see that the upper limit in biomass is linked to richness. In other words, you cannot go in French Polynesia, you will not have values above this because you're blocked here by the species richness available in that region. So the average regional biomass, that is, if you look here, French Polynesia, it's a value around here, and for New Caledonia, value around here, is dependent on this regional species richness. Now, if you look at total richness across the world, and we look at these alpha, that is reef diversity versus gamma diversity, that's the regional diversity, and biomass versus the reef diversity, we should have for two different regions. Here is, let's say, the center of the South Pacific, and here is Polynesia. We expect this, and this is what we observed. Now let's have a little look a little further into this graph. So here you have the number of species per transect, for instance, and the biomass on the same transect. For a given alpha diversity, you see that the biomass will be higher in the region with a low gamma diversity versus high gamma diversity. This is what we've already looked at. Now, if we looked at the maximum values of biomass, you see that in a region with low gamma diversity, the potential for biomass is lower than in a region with high gamma diversity. In other words, if you're in a species-rich region, your chances of having a high biomass is larger than in a region with a low gamma diversity. So now let's look at the last thing, that is production. Okay, we don't know really how to measure reef fish production at present, and this is mainly because we do not have values for fish mortality. So what we do, what's been done for quite a while, is what people have been calling instantaneous production. That is the production on a very short period of biomass during a very short period. So sorry for the equation there, but it's quite simple. P is what we call the instantaneous production for a given species. B is the biomass of that species, and delta T is a very short amount of time, let's say 1 day. So here is your alpha diversity and gamma diversity. Low, that's, let's say, Polynesian, and high, let's say that this is New Caledonia. So you know that the average weight is also linked to this alpha diversity, as we've seen before. You know that the biomass is linked in the same way. And the new thing here is growth rates. We know that the growth rate of small species is usually much faster than for large species, with exceptions, of course. There are some large species which grow fast, and there are some small species which grow slowly. But on average, this is what is found. So from this, we would guess that as alpha diversity increases, we would tend to have an increase in instantaneous production, but we don't know at present if low and high gamma diversity interfere with this relationship. So here is some true data again. So here you have French Polynesia and New Caledonia. Now you know quite well these two regions, and measures of, estimates of instantaneous production. So what you see here is first that it goes much faster in French Polynesia than in New Caledonia, and but also that the average instantaneous production in Polynesia is lower than in New Caledonia. So if we look at other regions in the same area, we see that the average instantaneous production is a kind of a function of the gamma diversity. But if we look at productivity, which is the ratio between this instantaneous production and biomass, we see that there's no difference. across regions. So what are some consequences of all this? Let's suppose that we have this relationship between biomass and local diversity for two islands. Island 1, which is, let's suppose, a small island with a rather low diversity, and Island 2, which is a large island or an island within a region where there's a very high diversity. And here is an example from New Caledonia, where you have the main island, which has a high diversity, versus Loyalty Islands, where the diversity is approximately twice less. And you do see that you do have indeed, for a given alpha diversity, a higher biomass on the small island versus the larger island. The small island versus the larger island. Okay, now if we look at the effects of fishing, we know that when you fish, you do decrease biomass by just because you crop the larger fish. But then you have a second effect, or a double trigger effect. That is when that's the effect number 2 here. That is, when you do fish, you do decrease what is called species density. In other words, which is kind of an assimilation of local diversity. In other words, you have less species truly available, which means that your potential for biomass decreases because of that. And then you have a third effect, that is, you tend to have smaller fish and smaller species as you increase the fishing effort. So this is a true example on the right, in gray you have a low fishing area, and in red a high fishing area, and you do see that you do indeed have this difference between in the relationship between alpha diversity and biomass according to fishing. Now, if we do look at our 4 regions, and you have in blue low fishing and in green high fishing, you do see that for biomass, you do have differences for only 2 regions, New Caledonia and Tonga. But for the other regions, we did not have, with the fishing levels we looked at, differences. Now, if we look at production, we see that there's no difference across regions. And if we look at productivity, we do again see that most regions are different. In other words, the productivity is higher in places where you're fishing. Why is this? This is because you have smaller individuals, and smaller individuals will produce more biomass per unit of biomass than larger ones. In other words, they grow faster. So one thing you should remember is that biomass is not a good proxy of production. Now, to end this talk, we're going to have a look at something which is very seldom discussed, is what we could call trophic Cudasack species. So what in the hell is this Cudasack species? If you look at the growth curve for what we could call normal species, you would have a relationship between the size and age, which looks something like this. Now, for these Cudasack species, you do have a very fast initial growth, then you reach a plateau and with very slow growth. And most of the reproduction starts from this inflection point. So if we look at 2 times intervals, T1, T2, which we'll call delta T1, and T2, T3, which we'll call delta T2, you see that the growth of the Cudasack species is much faster than the other species in the first time interval, but it's the opposite in the second time interval. So, which families provide species of this kind? Well, in particular, you have acanthurids, chaetodons, pumacanthids, tetraodontids, but you could also consider that most elasmobranchs are also Cudasack species, despite the fact that they do not have exactly this, they have a different type of growth curve. So here are some pictures of these species, just to make things more pleasant. So now if we look at a place where there's a high disturbance, the average age will be somewhere out here. If you're in a pristine area or in an undisturbed place, the average ages will be somewhere out there. So for the Cudasack species, once you're in an undisturbed area, you have mainly a production of gametes and very little somatic growth. Now, these species make nearly 70% of the biomass in undisturbed assemblages and nearly 0% of their somatic production. So undisturbed assemblages would be mainly exporters via across their gametes. And another thing that we know is that the recruits from these species are blocked to their access to the resources blocked by the adults, and therefore they usually cannot get into the system. So let's have a look again. If we do consider the same disturbed versus undisturbed and the contribution to production, what we would expect is that as you go from disturbed to pristine, that the Cudasack species would contribute more than the other species, and maybe that species-rich assemblages would be a bit higher than the species-poor assemblages in terms of contribution to production. So at present, we have not looked much into this, but the very few data we've looked at tends to confirm this, and that's you see that Fiji is a high gamma diversity versus Polynesia, which has a low gamma diversity, and you do see that indeed this contribution increases as you decrease fishing level. So this is all for now. If you have, please do not hesitate to ask questions, and please do not hesitate to use any of the information in this presentation. And if you need access to the original data, please write to me. I will be a pleasure to provide it. Thank you for your attention.
Thanks, Michel, for your excellent presentation. Now we open the space for the questions. Mari, you can open your microphone and camera, please. Pessoal, então agora que o Michel terminou, vamos então abrir a sessão de perguntas. Podem começar a colocar suas perguntas, quem tivesse alguma pergunta. E a gente vai aproveitar, então, fazer uma discussão aqui com a Mari sobre a apresentação do Michel para começar a engalar, começar a discutir os diferentes pontos que ele apresentou hoje, né? Mari, you can open or you can start your discussion here for the presentation for Michel.
Hey, thank you, Juan, for inviting me. It is always a pleasure to listen to Michel and all of his so interesting ideas. I've heard some of these before. I remember seeing some of these plots, part of these studies back in France. It's nice to hear you explaining to us all of the ideas behind those graphs, Michel. It was a very dense presentation, lots of ideas out there. Thank you.
Mm-hmm.
So I was, I think it's very interesting how you put the relationship between well, you can see for biomass and production with different drivers, and especially because you're trying to relate with the anthropogenic impact that we might see in reefs, because we know that what we see is probably biased. We might not be seeing in reefs the potential for biomass. When you were presenting those plots and you were comparing places that have low gamma diversity with locations that have high gamma and the biomass potential, I was intrigued because what first came to my mind is that when you are at a species poor location, then you don't have the potential to have greater biomasses than when you are in a species rich location. Could you comment on that? It was really intriguing.
So you have to be careful. The average biomass is lower in a species poor region than in a species rich region. Okay, so this means that if you go to, I don't know, some islands off Brazil, for instance, you'll have probably, on average, for a given fishing effort and so on, a lower average biomass than you would have in a species-rich region, let's say on the coast of Brazil. But if you had several islands and that you could plot the relationship between biomass and species richness, you would see that on isolated islands, very likely, I'm not certain that it works in Brazil, but it's just to give you an idea, we would expect to have a faster growing curve on these small islands than we do for the coast. Does not mean, as I was just saying before, that you would have a higher biomass on these small islands. It just means that the increase is faster, but the averages, as you probably notice, are a function of this gamma diversity as well.
Yeah, Michel, but this is one consequence. When you have the one small island, you have more concentrated biomass. For example, for UVC, in comparison for the coast.
Yes, well, the thing is that in most places, isolated islands are not exposed to fishing or other disturbances as much as places which are closer to shore. And this anthropogenic effect can be just huge. Well, I don't have graphs ready for you for that, but we know, for instance, that in pristine reefs of New Caledonia, we can reach biomasses, incredible biomasses in some places, over 1,000 grams per square meters. But how to say this? We do not have comparable things on the main island. Okay, so we cannot. It's very difficult because people say, "Oh yes, but when you go to these small islands or when you go to pristine areas, you see such huge biomass." So what you're saying is probably not correct. Well, actually, the problem is that it's extremely difficult to compare places where the fishing levels are so different. For instance, for sharks, which make quite often a rather large proportion of the biomass on these isolated reefs, they're nearly extinct on the major islands, and this just that makes a huge difference.
Cool.
Same thing for the very large groupers, well, the large rays. Most of the large fish just disappear very quickly with fishing. And this has been illustrated over and over. But if you take places with similar disturbance levels, I'm pretty sure you should find approximately the same type of results wherever you go. But as I was saying earlier too, is this what I presented today is preliminary. It does not mean that it works everywhere, but we see these curves in the places we've been, and it's up to other people to check if they find the same results elsewhere. If they do, then we do have a good signal. If they don't, we have to know why. In both cases, it's a win-win situation.
Yeah. Michel, I have very curiosity because you showed all the time the result for the gamma and alpha diversity in correlation with biomass, but you think is pertinent you comparing, for example, beta diversity on different components for beta diversity and your correlation and with biomass, for example. I suppose the size you don't have more anthropogenic effects. Probably you have more turnover for the species, but the biomass is not necessarily responding these changes.
Okay, I don't know too much about the role of beta diversity in all this. As a guess, I would say that when you increase beta diversity, you would tend to increase the biomass for a given level of species. But this is just a guess. I don't. I may have something done on that, but it's been long time ago, and I wouldn't be able to. I'm not so certain. One thing which is kind of interesting is that if you look, we talking about species richness essentially, but you could use what we called, in quote, functional richness, and you would find the same things. For instance, Camillo Mora did an article with functional richness, and he did find exactly the same type of curves on a global level.
There's a question from
you
Gabriel.
You can read, please.
Michel, there's a question from Gabriel Garcia.
Yes.
I don't know if you can read it, but I'll
No, I don't see it. That's a problem.
Okay, I'll read it too. He says it was a nice talk, and he was wondering what you meant with reef fish functions, quotes. He's asking, are those ways in which fishes may alter their habitats, or is it based on resource usage?
All right. So, as I was saying earlier, at present we have a very limited view of what we could call true functions. For instance, a lot of people say herbivory is a function. Okay, that's okay. I'm fine with that. But then when you look at the herbivory, you have all kinds of different ways to eat algae, and depending on the way you do it and which algae you do eat, you could think that these are different functions. But at present, we do not have enough information on the diets, on the behavior, on of the fish altogether. So we know that we cannot truly reach these ecological functions. So what we do is we use proxies, and these proxies, the most common proxy is to use a combination of life history traits. Okay, so this is very debatable. Bellwood had an article in 2019 which criticized this approach, but he did not propose anything different. So at present, we're kind of stuck with this way of doing, which is, by the way, used in many other fields. And for instance, in botany, for birds, for mammals, they use exactly the same type of approach, because it is extremely difficult to really know which true ecological functions exist out there.
Thank you, Michel. So, can you do that, or would you like to? You can go ahead.
One question and another question is only for more dynamic. Well, you have another question here for Sergio Flotter. Is the plantivores are Cudasack species?
Pardon? Oh, if plantivores are Cudasack species, some of them could be. If you take, for instance, the manta rays, you could think of them as Cudasack species. But in general, I cannot imagine in teleosts. Well, at first think I cannot. I don't see any in the teleost, but in the elasmo branch, yes. But it's just a matter of looking. But they are probably within the plantivores. I would say they're probably rare, if there are any. they're more into the herbivores or piscivores or carnivores.
Cool. You have another question here for Christiane Harboy. Very nice talk, Michel. Congratulations. I work house microbiota interaction. I use it to investigate the alpha and beta diversity, but gamma is new for me. Could you please explain me a little bit?
Okay, so diversity is a matter. There's a matter of scale. Okay, when we talk about alpha diversity, I mean, at least in this presentation, truly, we're not talking really about diversity. We're talking about species density, which means that it's the number of species for a given surface. Then, when you go to larger area, then most people will tell you there's so many species within this island, there's so many species within this region. So crudely, we speak about island diversity or regional diversity, which are in both cases called gamma diversity because they contain all the species found into subdivisions of what they cover. I don't know if this makes sense or not. But the problem is that given when you're comparing islands do not have necessarily the same surface. Regions do not necessarily have the same surface either. So one approach to this is to look at the number of species per geographical cell. This is what you've seen on the maps earlier, which makes abstraction of the problem of island area or regional area, and which allows you to compare regions or to compare islands in terms of their gamma diversity, which is something we did not talk about today. Does that answer the question?
Yes, I think so. Thank you, Michel. Karini Emer. Hey, Karini. Thank you for your nice talk. And she was wondering how she was thinking about different components of different types of diversity. And she's asking, what do you expect to find if you expect to find the same relationship between phylogenetic diversity and productivity, or functional diversity and productivity?
Well, I don't see why phylogeny would. I'm not certain I understand well the question. That's the thing. But okay, we've been talking about species from a taxonomical point of view. We could group species into higher orders of taxonomy, such as genera or families or whatever, or in some cases, but rare cases, in terms of populations, the other way down. I don't think it would change much. It's just a matter of grouping the species in different ways, which I have a guess, but it's just a guess, that when you make up these functional groups, or if you look at taxonomical groups, you would end up somehow mathematically doing the same thing. And in other words, I would expect that taxonomy and functional groups would tend to have the same results, in particular because within a genus or within a family, you usually have a convergence in life history traits. But I don't really know if this answers the question or not.
Yes, I think that if you do that for for functional groups, when you gather species that belong to very distantly related phylogenies, then you might have something that would be different from what you see at the family level for the relationship between species richness and biomass, but I don't think that for phylogeny you would expect something different. Do you want to go on, Juan?
Yeah, okay. Michel, do you data grade for the recently paper for Renato Moraes of the production? How did you opinion your more recently paper for Renato is calculated?
Oh, yeah, I read that article quite a while ago when it came out, but unfortunately, I'm sorry to say that my memory is not so good, so I have to, let's see if I can find it quickly in my browser to have a recall of what's in there. Sorry, I don't have instantaneous recall of this. Where could it be?
Yeah, Renato tried to estimate more the production using the biomass, observing the UVCs, and use the different equations.
The different what?
Mathematical equation for trying to calculate the production using, for example, the somatic growth and age and the mortality.
Yeah, the problem is mortality. Okay, in most cases, we do not have access to true mortality for, I would say, 95 or 99% of reef fishes. We have extremely little data there, true data. So what people use is proxies built on the relationship between species size and mortality, or species growth curves and mortality. But these are only proxies. If you look at because fish on an exploited reef are submitted to fishing, and fishing will affect differently species, of course, but also from one place to another, you will not have the same impact of fishing for numerous reasons, because they don't use the same gear, because they don't use it in the same way, because of food preferences, of you name what. So in my opinion, the mortality rates which are used so far are just indicative, and it's, in my opinion, it generates values of production which are indicative. I have no problem with that, in terms that our instantaneous production estimates are also very questionable, okay, because the growth rate of species is not the same from one place to another. It may change from one year to another. We do not see all the individuals when we do reef fish counts, in particular the very small individuals which have the fastest growth are quite often not observed, either because they're hidden in the reef or because they live in different environments. Therefore, we have to be extremely cautious about all these production, or either instantaneous or total production estimates for reef fish. But the major thing is comparative approaches. That means that as long as you're comparing one place to another, you may end up saying, or be able to say, that production is higher in place A than in place B, with a certain confidence, which is already a very interesting result, even though we know that the values we're getting for these productions are probably wrong. But in relative terms, they're probably okay.
Thanks, Michel. Well, people, Michel, thanks for your talk today, and this discussion is very important. Put these different media in the table today. Thanks again. Pessoal, muito obrigado a todos, Mari, por ter participado. É muito legal ter também você aqui para ajudar na discussão e fazer mais enriquecedora essa discussão aqui hoje. Então, pessoal, eu quero convidar todos vocês para nosso próximo CEBIMário, que vamos continuar com o pessoal de fora. Vamos convidar, então, a próxima semana o Dr. Rui Rosa, que vai falar sobre impactos biológicos de aquecimento global, acidificação e dessodinização em hábitats marinhos. Então, espero todos vocês a próxima semana para participar também desse seminário que provavelmente será em português. Não sei ainda. Mas, thanks, Michel, and see you after.
You're welcome.
Bye-bye.
All right. Bye-bye. See you later.